Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Welcome to the deep dive. We take the sources, you get the knowledge. Today, we're digging into something really common and honestly pretty debilitating: tendon injuries. Just picture this. You hurt your hand, maybe one of those flexor tendons, and you need surgery. Okay, that's tough enough.
Right.
But then imagine finding out that almost a quarter of people in that situation, up to 24%, end up needing a second surgery.
Yeah, just to deal with scar tissue.
Exactly. Scar tissue that's painful and stops them from moving properly. We're talking hundreds of millions just for hand surgeries like this every year in the U.S. alone. It's a huge issue. And beyond physical therapy, there haven't been many answers.
It's true. And for the longest time, tendons were seen as, well, kind of simple, like biological ropes, basically. Strong, sure, but not very dynamic at the cellular level.
It's just connectors.
Pretty much. But that view is really changing. New research is showing just how incredibly complex they are, especially when it comes to healing.
And that's exactly what we're diving into today. We've got this really groundbreaking study from the University of Rochester. It's genuinely shifting how we understand and potentially treat these injuries.
Absolutely.
We're going to unpack how scientists are now targeting specific cells to try and cut down on that problem scarring. This could honestly transform recovery for so many people.
It really could. So the first thing to grasp is just how far our understanding has come. Like you said, for decades, tendons were just seen as these straightforward sort of rope-like things.
Functional, but maybe a bit boring biologically.
Kinda, yeah.
Yeah.
But the work coming out now, especially from places like Rochester Center for Musculoskeletal Research, paints a totally different picture. We're seeing tendons as these highly complex structures.
With intricate healing processes, you mean?
Exactly. They're finding this surprising diversity of cells inside the tendon, and crucially, figuring out how these different cells talk to each other, how they interact after an injury, sometimes helping, sometimes hindering.
So it's not just one cell type doing everything, and their actions aren't always beneficial. That sounds like it makes healing tricky, especially when a tendon is completely severed.
It does. When you have a severed tendon, say in the hand, the standard approach is surgery to stitch it back together. But the healing, often, it's not great.
Why is that?
Two big reasons, really. First, the tendon tissue itself isn't great at regenerating new cells. It's a slow, weak process. Second, there's a really high chance of forming excessive scar tissue. And we're not talking about the good kind of scar that holds the repair.
So this is that problematic scar tissue that leads to the second surgery, the stuff that forms around the repair site.
Precisely. You need some scar tissue right between the repaired ends to bridge the gap. That's essential. But the real villain is the excessive scar tissue that builds up around the outside of the tendon, called peritendinous scar tissue.
And that's what gums up the works, restricts movement.
That's exactly it. It can severely limit how much you can bend your finger or move your limb. And right now, apart from Villainy doing physical therapy, there's no specific treatment after surgery to stop this bad scarring from happening.
Okay, so tendons are complex, healing is tough, and this external scarring is a major roadblock. This leads us right to the new study. How did they pinpoint a key player in all this?
Right, this is where it gets really interesting. For years, the research focus was mostly on tenocytes.
These are the main tendon cells, right? The ones that build the structure.
Exactly. They build and maintain the tendon matrix. Important cells, definitely. But this new work, led by Assistant Professor Ann Nichols, looks somewhere else at a group of cells called epitenon cells.
Apetinon cells. Okay, that's probably a new term for a lot of listeners. What are they?
They form a very thin layer, kind of like a sheath, that wraps around the entire outside of the tendon.
And why were they overlooked before?
Well, people knew the epitenon was involved early in healing. It was seen as maybe an orchestrator, possibly contributing to collagen, and maybe even those scar-promoting factors. But the problem was, scientists couldn't specifically target just those cells. They couldn't isolate them easily to study exactly what they were doing.
So they were hard to track. How did this team get around that? This must be where their clever methods come in.
Yes, exactly. This was the first big hurdle they cleared. They needed to find a way to specifically label these epitenon cells. They searched for a unique genetic driver, think of it like a unique tag or barcode for these cells.
And they found one.
They did. They found that a gene called GLAST, that's glial high-affinity glutamate transporter, was a pretty specific marker for these Apetinon cells, and importantly, it worked in both mice and humans.
Okay, so they found the tag. Now they could put a spotlight on these cells. What next?
With that tag, they used a really neat technique called genetic lineage tracing.
Sounds complex. What does that let you do?
It essentially lets you follow the descendants of those tagged cells. So they could tag the guialast expressing Apetinon cells at the start, injure the tendon in mice, and then literally watch where those cells went and what they turned into during the healing process, like a cellular GPS tracker.
Wow. And they also use something called single-cell RNA sequencing.
Right. That's another powerful tool. Think of it as zooming in even further. Instead of just tracking where the cells went, this technique let them see what each individual cell was doing at the genetic level.
Like reading its instruction manual at that moment.
Exactly. Which genes were turned on, which were turned off. It gave them a really detailed molecular signature for each cell's behavior. Was it building tendon? Was it promoting scarring? Something else.
Okay, so armed with these tools, the tracker and the molecular snapshot, they could finally see what these Apetinon cells were really up to. And they found this kind of split personality.
That's a great way to put it. What they discovered was pretty fascinating. These Apetinon-derived cells, the ones they tracked, essentially had two different fates. And crucially, their role, good or bad, depended entirely on where they ended up after the injury.
Location, 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 even at the cellular level. So what were the two main locations and what happened in each?
Okay, so some of these Apetinon cells migrated into the gap between the severed tendon ends. This area is called the bridging tissue. It's the crucial stuff that reconnects the tendon.
And what do they do there?
In that bridging tissue, they actually differentiated, they matured into proper tenocytes, the good guys, the tendon-building cells. They were actively contributing to regenerating functional tendon tissue.
So helping with the actual repair.
Exactly. In fact, they calculated that by day 28 after the injury, about a third of all the new tenocytes in that crucial bridging area had come from these Apetinon cells. That's a significant contribution to good healing.
That sounds fantastic. But you mentioned a split personality, so not all of them did that.
Unfortunately, no. A larger percentage of these same Apetinon-derived cells took a different path. They migrated to the outside of the injury site.
Ah, is this where the problematic scarring comes in?
Precisely. These cells formed a thick capsule of scar tissue around the periphery of the repair. This is that unwanted, function-limiting paratendinous scar tissue that causes so many problems with movement later on.
Wow. So the same starting cell type can become either a healer or a scar former just based on where it goes. That's incredible. Did they figure out when the scar formation was most active?
They did. They pinpointed a specific time frame, a kind of fibrotic burst, between days 7 and 10 after the injury. This seemed to be the critical window when those peripheral scar-forming Apetinon cells really ramped up their activity.
Okay, a critical window. And did the genetic signatures from that RNA sequencing match these two different roles?
Yes, perfectly. The cells that formed that outer scar capsule showed high levels of genes associated with inflammation and fibrosis, pro-inflammatory and pro-fibrotic genes. Think genes with names like CXC motif chemokine ligand 14, peptidase inhibitor 16, and interleukin 6. These were clearly driving the bad scarring process.
And the ones that went into the bridging tissue?
Those cells, in contrast, were expressing genes linked to tenogenesis, making new tendon, and organizing the extracellular matrix, which is the structural scaffold of the tissue. Their genetic profile clearly pointed to a pro-regenerative, constructive role.
So you have these clear genetic signatures distinguishing the good cells from the bad cells, even though they started from the same place.
Exactly. And knowing which cells are causing the problem, where they are, and when they're most active, that's the absolute key for designing a targeted therapy.
Which is exactly what they did next, right? They used this knowledge to try and intervene. What was the strategy?
It was quite elegant, actually. They used genetic engineering in mice so that specifically those guialast-expressing Apetinon cells could be triggered to undergo apoptosis that's programmed cell death.
So a self-destruct switch just for those cells?
Pretty much. They could activate this switch by giving the mice a specific substance, diphtheria toxin, but only the cells expressing guialast would be affected.
And they targeted that critical window, days 7 to 10.
That's right. They waited until that fibrotic burst window and then injected the toxin directly at the injury site in the mice's paws. The goal was to selectively eliminate the Apetinon cells that were destined to form the problematic outer scar without harming the ones contributing positively to the bridging tissue or other important cells.
Okay, the moment of truth. What happened? Did it work?
The results were really striking. The mice that got this targeted treatment showed a significant improvement in the range of motion of their little toe joints, essentially, how far their fingers could bend.
How much improvement?
Quite a bit. For instance, at 28 days after injury, the treated group had an average range of motion over 60 degrees, while the untreated control group was stuck around 46 degrees. That's a big difference in function.
That's a major gain in movement. Did they see other benefits?
Yes, they also measured gliding resistance. Lower resistance means the tendon slides more freely.
Less friction, less catching.
Exactly. And the treated mice had significantly less gliding resistance, which is direct evidence of less restrictive scar tissue formation around the tendon.
Okay, less scarring, better movement. But what about the strength of the actual repair? Did removing those cells weaken the tendon? That would be a concern.
That's a critical question, and the answer was very encouraging. They tested the mechanical properties, the maximum load the tendon could handle before breaking, and its stiffness. And crucially, there was no difference between the treated mice and the untreated mice.
So they reduced the bad scarring and improved movement without compromising the strength of the healing tendon itself.
Exactly. That's the ideal outcome. You get the functional benefits without the structural drawbacks.
That really does sound like the holy grail for tendon repair. Less scar, better motion, same strength. Now, the huge question, does this translate? Can we apply this to humans?
That's obviously the ultimate goal, and the researchers took a big step in that direction. They didn't just rely on the mouse data. They got samples of actual human scar tissue, the peritendinous kind, from patients who were undergoing that second surgery, tenolysis, to remove it.
And they compared the cells in that human scar tissue to the problematic cells they identified in the mice.
They did. They looked at the gene expression profiles, and what they found was, in their words, a remarkable transcriptional similarity. The problematic scar-forming epitenon-derived cells in the mice looked genetically very similar to the main scar-forming cells called fibroblasts found in the human scar tissue.
So the same kinds of genes were active.
Yes, and they even pinpointed a specific gene marker called CCN3, that's communication network factor 3, which was highly expressed in the bad Apetinon cells in mice and in the fibroblasts from the human scar tissue samples.
Wow. That's a strong link.
That's a very strong link. It strongly suggests that the cells causing the problematic scarring in humans likely also originate from that Apetinon layer, just like in the mice.
Which means the findings are likely translational, applicable to people.
Highly likely, yes.
Wow.
That's the exciting implication. If we can develop strategies, maybe drugs, maybe other targeted therapies, to specifically modulate or even remove these particular Apetinon-derived cells in humans during that crucial 7 to 10-day window after surgery.
We could potentially prevent a lot of that excessive scarring.
Exactly. It could significantly reduce scar formation, lead to much better range of motion, less pain, and hopefully dramatically reduce the need for those difficult, costly second surgeries. It really opens up a new avenue for treatment.
This has been a fascinating deep dive. It really drives home that tendons are anything but simple ropes. This study feels like a major step, giving us the first really detailed map of how and when these Apetinon cells influence healing.
And it highlights this really interesting biological concept, these bifated cells. Cells that can go down two very different paths, healing or scarring, depending on the local environment and the timing.
Which implies that just trying to block all scarring might be too blunt an instrument.
It does. It suggests we need much more specific, localized interventions, precisely timed, like the approach they tested. It's about being selective, not just stopping everything.
And it also really shows the power of combining these cutting-edge lab techniques like the lineage tracing and single-cell sequencing with studies that actually look at human patient tissues.
Absolutely. That combination, bridging the gap between animal models and human samples, is critical for speeding up the process of getting discoveries from the lab bench to the patient's bedside.
Now, like all good science, this study probably has some limitations or areas for future work, right?
Of course. And the researchers are upfront about them. For example, that GLAST marker they used, while useful, wasn't perfect. It only labeled about a quarter of all Apetinon cells. And it did label a few other cell types, too.
Yeah.
So the hunt is still on for even more specific markers.
So refining the targeting tool? Anything else?
Well, this study focused on what happens after the injury starts. It didn't really dig into the very, very initial signals right after the trauma that first wake up these Apetinon cells and tell them what to do. Understanding those earliest triggers could potentially offer even earlier points for intervention.
before that fibrotic burst even starts.
Potentially, yes. And one other important point they raised is that tendons aren't all identical throughout the body.
Ah, so what works for a hand tendon might not work exactly the same way for, say, an Achilles tendon.
It's possible. Different tendons might have slightly different cell populations or respond differently to injury. So while this study provides a fantastic foundation, more research is needed to understand this diversity across different tendon types. This is a huge first step, but it opens many more doors.
It really does. So to wrap up, we've journeyed from seeing tendons as simple ropes to understanding this complex cellular drama starring these Apetinon cells with their dual identities. By figuring out how to selectively target the ones causing bad scarring, this research shows a potential path to vastly improved healing after tendon surgery, potentially saving countless patients from needing that second operation.
It's a really hopeful piece of work. And it underscores a key idea in science, doesn't it? Sometimes the biggest advances come from taking a closer look at something previously overlooked, like this thin little layer of cells around the tendon.
It absolutely makes you think, if we're just now unlocking these secrets in tendons, what other parts of the body that we consider simple might be holding similar complexities? What other common problems might have these incredibly precise cellular level solutions just waiting for us to find them?
It's a great question. The potential is enormous.
Definitely something to ponder. That's all for this deep dive. Thanks for joining us.
Thanks for tuning in to the Health Headliner podcast by Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.